realized I don't have to have the lights down for the screen. As I said before, if you're taking notes or you're trying to follow along, having the book open for this particular chapter, I'm kind of going in the same order that the book goes. It's kind of like having PowerPoints. This section is on early development of cancers. Cells get damaged, especially as you age, is that the cells, the telomeres wear down, remember? And the cells don't want to repair themselves as well. They don't want to make more copies. They don't want to repair the tissues. And you get these, what are called precancerous lesions. Groups of cells that could become cancerous but aren't quite there yet. Polyps along your digestive system. What they call breast in situ, where they take a lump out and it's not cancerous, but it's not, it could have developed into cancer. What they found, or what they have found, is that by rolling the mammogram, the common repeated mammograms to an earlier age, they are finding more in situ in women, taking them out. But long term, not really, there's no real indication that it's changing mortality from breast cancer. And that's kind of a mystery unless you realize that a lot of those little changes, we're not going to progress to breast cancer. These cells are damaged. They're more likely to die and the lumps to kind of disintegrate than for them to go on to become full-blown tumors. And this happens a lot of places. It happens with colonic polyps. It happens to some extent with the differences in pap smears. Pap smears are different because the changes are viral induced. And when you get a precancerous pap smear, the issue there isn't are the cells going to progress to cancer? It's that the changes are not necessarily just from the viruses. Sometimes there's other parasitic infections in the female system that can produce similar changes in cells that would never progress to cancer because that's not what's going on. Changes in the esophagus sometimes can be picked up and are precancerous lesions that may or may not progress to cancer. There are what are called driver mutations early on in the development of the cancer. As cells start to be more and more damaged, there's certain changes that can happen, and when they do happen, it tends to really push the system toward becoming cancerous. Remember, there's potentially many, many, many different mutations that have to happen. And if you do the odds of them accumulating in a human lifetime seem pretty long, but driver mutations will push the system along through what are called hypermutation events and produce a lot of the changes that would make the cells become legitimate cancer cells. Now, once you've got a population of legitimate precancerous or cancerous cells, in a lot of ways, this is almost like a colonial organism. They may not be differentiating into the cells they're supposed to be, but it doesn't mean they're not different. It doesn't mean that mutations and abilities are showing up in some cells that are not showing up in other cells, and that the clump of cells in the tumor may be helping each other out and supporting the tumor like a colony of eukaryotic cells would. I've got a question. Is it useful to consider this a parasite? It does progress and evolve like an organism in an ecosystem. But I'm not sure that it is useful to look at it like it was actually a parasitic entity. Because there's other interactions that parasites don't necessarily do. As the cells divide, they tend to get more damaged. The damage produces more variation, more different cells, and of course what's happening is selection. Is cells that are damaged in ways where they can't survive, they're gone. Cells that are damaged in ways where they're easily recognized by the immune system and removed, they're gone. cells that can't continue to progress stall. Sometimes you have cancers that produce tumors. Brain tumors very rarely progress beyond a certain point. They're making a big lump of cells inside your head, so it can be dangerous. But metastasis is actually more of a rarity in brain tumors than in a lot of other tumors. This lump of cells isn't just cells. It's a lot of material is being extruded, and there's a matrix called a stroma inside a tumor. And that's interacting with the cells. That's interacting with the surroundings. That, in some cases, is making it difficult for the immune system to access the cells. One of the things that happens very commonly in cancer, early cancer cells, is a reliance on glycolysis as a source of energy, right? The first part of respiration, the non-oxygen part of respiration. And also, it ties early cancers much more to glucose as an energy source than breaking down, say, lipids or proteins for energy. It's not an across-the-board thing that can be relied on, but some people have thought to go on very low sugar diets, very low carb diets when cancer's in an early stage and sometimes in a later stage, to try to deny it its preferred fuel. That's, again, they're still kind of working the bugs out of whether that's a legitimate approach or not. What does also happen is as the tumor grows, it needs oxygen and nutrients if it's going to go into aerobic respiration. And the cells all the way in the middle are a long way from oxygen and nutrients. So there's a problem that needs to be solved, and there's two basic ways that tumors tend to solve this. The first one is happens where there is a gene that activates, and it produces what's called angiogenesis. When you're a growing kid, you're adding new tissue, you need that tissue to be supplied with blood vessels. So there are proteins released that basically say to nearby blood vessels, give us some extent, give us some capillaries, give us some extension down into this tissue, we need to supply it with blood. A lot of tumors will activate that protein, which would not be activated typically in an adult because we've got all our blood vessels. And there's a type of chemotherapy that is tries to counteract that. We'll starve the tumor cells of oxygen and they'll just die, and it'll help get rid of the cancers. And it seemed like a really good idea. They started this like 20-some years ago. And it's only moderately effective. That's because the cells can also activate oxygen debt, where they can just use glycolysis to provide the ATP that they need. They're not high metabolism cells, so they can kind of get by. If the middle of the tumor is oxygen starved, it will produce lactic acid. That in itself can have effects, but also it's hiding from your immune system, right? Your immune system is supposed to be able to recognize damaged cells and cancerous cells. For cancers to survive, it needs to somehow counteract that. In some cases, it produces chemicals that make recognition difficult. In some cases, it produces chemicals that reduce cooperation and communication amongst the immune system. Basically saying, nothing to see here, go away. Don't talk to those cells over there. It's all fine. Now, as the tumor grows, I tend to do this. It's not a really smooth-edged object, especially as it gets more advanced. It tends to invade local tissues with little tendrils. The edge gets very uneven. And in fact, the term cancer comes from, in ancient Greece, when they would remove these lumps from people, they would find they weren't just set lumps. They had these, what looked like crab legs sticking out of them, cancer of the crab. And so there's what they call margins. If you have some sort of lump that needs to be taken out by a surgeon, they will take the lump and send it to the lab where my daughter works, and they'll mark it it's bigger than just the lump. They take some extra tissue. They take a slice through and see whether there are microscopic extensions beyond the margin that they cut. And then the message would go up to the operating room, take out more. Is that this thing has extended beyond the what you've excised. So margins are really important. It hasn't metastasized yet, but you haven't gotten it all. So it's gonna keep developing if you don't get it all. Okay, that's the end of this particular